Picking up with our second video for Chapter 5, we're going to start talking about diversity in polymers. Remember that each cell has thousands of different kinds of macromolecules. You're going to want to make sure that you have those four major macromolecule groups clearly down, cemented in your memory banks. Macromolecules will vary between cells within a single organism. They'll vary even more within a species and even more between species. So just really, really, truly diverse in their nature. Here's a whiteboard question. You want to be able to name those four macromolecules. So you should be able to do that. Boom, 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 boom. In the classroom, I sometimes divide the room in half, tables 1, 3, 5, tables 2, 4, 6. If you're playing along in the online version, just go ahead and answer both sets. But you want to be able to list the monomers for both of these. No, we actually haven't said this in class. It was on your initial slide, but this is also one of the things I do in class is to see if you've been reading ahead at all, to see if you're staying in front of me in the classroom the way you're supposed to. So let's actually head into that area of topic. So carbohydrates. We'll start as carbohydrates. Carbohydrates serve as fuel. They are building materials. They're classified based on the number of simple sugars that they contain. So carbohydrates are polymers. They are polymers, and the monomer of carbohydrates are sugars. The simplest carbohydrates are single sugars. They're called monosaccharides, mono being one, so monosaccharides. The three that we deal with are glucose, fructose, and galactose. We can also have disaccharides and polysaccharides. Okay. So we'll talk a little bit more about those terms in a little bit here. But remember that carbohydrate macromolecules are complex carbs. Those are polysaccharides, they're polymers of simple sugars. So macromolecules of carbohydrates are going to be things like starches and fibers. Simple sugars, as I said, are going to be the monosaccharides. They're going to have some multiple of one carbon, two hydrogen, and one oxygen. So some multiple of that. So glucose, for example, is C6H12O6. I can take each of those in the top bullet and multiply by 6 and get C6H12O6. Glucose being the most common monosaccharide, it's the one that our cells can actually utilize. The job of simple sugars, store energy in their chemical bonds. We're going to see sugars play a major role in photosynthesis and in cellular respiration. They'll also function on a skeletal level. They're a raw material for the synthesis of other monomers, including those for amino acids that we'll see when we talk about proteins, and for fatty acids when we talk about lipids. One of the things that you'll want to just kind of store in your memory banks, sugars typically end in ose. See this? Glucose, fructose, galactose. You've heard of lactose, sucrose, any of those guys. So if you see a word, you see a macromolecule and it ends in ose, good first guess is that it's a sugar. Okay? It's a great place to start in your guessing game. These are disaccharides. Remember what we said, that di stands for two. Okay, di and bi will both mean two, but in this case, when we talk about sugars, we're talking about disaccharides. Disaccharides happen when we have a dehydration reaction. Dehydration reaction that joins two monomers. And what ends up happening is we get a specialized bond called a glycosidic linkage. Okay, so we're going to lose, just like any other dehydration reaction, we're going to lose out this water, and we're going to get this specialized bond right here called a glycosidic linkage. That's important when we talk about disaccharide formation. And then we're going to take a look at polysaccharides. Polysaccharides are polymers of sugars. So I have a whole bunch of monomers and monosaccharides and disaccharides that get put together, and I get a polysaccharide. Polysaccharides are used for storage. We talk about starch and glycogen in mammals. We talk about cellulose and chitin in the invertebrates and in plants. and they have very different structural roles. So, right? So we store sugars as starch and glycogen in you and I, but in plants, we actually take those polysaccharides and they have a more structural. Cellulose being one of the primary components in the cell wall of plants, chitin being the primary component in the exoskeleton of some of the invertebrates. So structure and function of polysaccharides are really determined by the sugar monomers and the positions of those glycosidic linkages that we just talked about. Here is that separation of starch and glycogen being the storage polysaccharides. So starch we're going to see in plants. I think I said that wrong on the last slide, didn't I? Sure did. I was talking about animals. Sorry about that. So starch and glycogen are the counterparts to one another. Starch being the one that we see in plants, glycogen being the storage polysaccharide in animals. So plants are going to store surplus starch as granules. They do that within their chloroplasts and other plastids. We'll talk about plastids and chloroplasts when we get to the cell chapter. That's coming up next, I believe. Glycogen is how animals will store polysaccharides, and glycogen is a storage unit that we use predominantly in the liver. You'll also see it in muscle cells, but the big place that we store it is in the liver when we convert excess glucose into glycogen, and then it gets stored in the liver so that we can later on utilize it, break that polysaccharide back down into glucose and have it enter the bloodstream. Okay, so those are our storage polysaccharides. I believe our next ones talk about structural polysaccharides. So again, here's cellulose. The polysaccharide cellulose is a major component in that tough outer cell wall of plants. Like starch, so very similar to starch, but like starch, cellulose is a polymer of glucose. The difference being how the glycosidic linkages come together. So remember that's the big thing is the glycosidic linkages. And really the difference is based on two different forms, ring forms of glucose, an alpha ring formation versus a beta ring formation. A tiny little side note here, enzymes that digest starch by hydrolyzing its alpha linkages are not able to, those same enzymes are not able to hydrolyze beta linkages of cellulose. So the enzymes that you and I have that allow us to break down starch because of its alpha linkages do not work on cellulose because there's a different shape to that molecule. So what ends up happening is cellulose becomes biologically inactive in humans. We don't have the enzymes we need to break it down. So we use it as fiber. Cows, termites do break down cellulose. They actually have a gut bacteria. Cattle have a gut bacteria that make it work, and there's a different enzyme reaction that happens because of that bacteria that allows them to utilize the cellulose. We're going to talk about chitin, and then I think I've got one whiteboard here, and then I think we'll move on. But chitin is another structural polysaccharide. As I mentioned, it's found in the exoskeleton of arthropods, insects, spiders, crustaceans, those guys. It provides structural support to the cell walls of many of the fungi. So there's in medicine, we have a suture material made out of chitin. It's called proline. So if any of you have ever had sutures and they used proline, it's made out of chitin, which I think is pretty cool. But again, so chitin is another structural protein like cellulose. And then two questions on this whiteboards, and then I think I've got one more whiteboard slide. Monomers, what are the monomers of carbohydrates? And then name the covalent bond that creates a disaccharide. What is the name of that type of covalent bond? Name the storage carbohydrate in animals, the storage carbohydrate in plants, and two structural carbohydrates and where they might be found. So I want to wrap that with carbohydrates. Next up, we'll start talking about lipids. Good place to break. This video was a little bit longer. We're almost hitting 10 minutes, so I'm sorry about that. Back with lipids.